Good morning, students. In this class, we shall discuss one mark questions on the lesson, that is molecular basis of inheritance. First question: name the pentose sugar present in DNA. The DNA stands for deoxyribose nucleic acid. The pentose sugar present in DNA is deoxyribose. Second question: name the pentose sugar present in RNA. The pentose sugar present in RNA is ribose sugar. Because of the presence of ribose sugar, it is called as ribose nucleic acid. Third question: mention an example for purine. So the purine and pyrimidine together, it is called a nitrogen bases. So the nitrogen base, phosphate group and the pentose sugar, all these three together are going to form a nucleotide. Okay, so this nitrogen bases, there are two purines and two pyrimidines. So here they are asking one example for purine. So how many purines are there? Two purines are there. What are those two purines? Adenine and guanine. As here they are asking only one purine, so among these two any one you can mention. Fourth question: mention an example for pyrimidine. So I said nitrogen bases are of two types, that is purine and pyrimidines. So two purines and two pyrimidines. But when you have studied the difference between DNA and RNA, so in the DNA two nitrogen base, in the DNA there are two purines and two pyrimidines. In the RNA also there are two purines and two pyrimidines. But what the pyrimidine is present in the DNA, that pyrimidine is absent in case of RNA. Instead of that absent pyrimidine in the RNA, it will be replaced by one more pyrimidine. Okay, so in the DNA, what are the two pyrimidines present? So in the DNA, cytosine and thymine are the pyrimidine, whereas in case of RNA, there are two pyrimidines. What are those? Cytosine and uracil. So instead of thymine, uracil is considered as a second pyrimidine. So how many pyrimidines are there totally? So there are three pyrimidines, that is cytosine, uracil and thymine. Among these three any one you can mention. Name the bond present between pentose sugar and nitro base in a nucleoside. The nucleoside is consisting of pentose sugar and nitrogen base. Those two are binded with the bond, or the linkage is called N-glycosidic linkage. Sixth question: name the bond present between a nucleoside and the phosphate group. Nucleoside and the phosphate group are combined together and forms the nucleotide. So that nucleoside and phosphate group are binding together to form nucleotide with the help of phosphoester linkage. Seventh question: name the bonds present between the two strands of a DNA molecule. The two strands of the DNA molecule is having the presence of hydrogen bond. Eighth question: name the bond formed between sugar and phosphate backbone of single stranded DNA. So in between sugar and phosphate, that is the backbone of the single stranded DNA. So there in between them there will be presence of phosphodiester bonds. Ninth question: What is alternative chemical name of thymine? So thymine is also called as 5-methyl uracil. Next, 10th question. Name the nitrogenous base present in DNA but not in RNA. As previously I said, there are some differences between RNA and DNA. One among them is the presence of nitrogen base, whereas in case of DNA there will be presence of thymine, and that thymine is not present in RNA. 11th question. Name the nitrogenous base present in RNA but not in DNA. RNA is consisting of two pyrimidines. What are those two pyrimidines? Those two pyrimidines are cytosine and uracil. So here uracil is present only in RNA. It will not be present in the DNA. 12th question. Name the nucleoside present only in DNA. The nucleoside present only in DNA is called deoxythymidine. It is also called as even uridine. Next, 13th question. Name the nucleoside present only in RNA. The nucleoside present only in RNA is deoxyuridine. 14th question. What was the observation of Erwin Chargaff for double-stranded DNA? The Erwin Chargaff observed about the double-stranded DNA, and he stated that the ratios between adenine, thymine, guanine and cytosine are constant and equal. 15th question. The two strands in a DNA molecule are said to be anti-parallel. Why? The two strands of the DNA are said to be as anti-parallel. One strand, if it is running in the direction of 3' to the 5', another strand of the DNA will be running in the direction of 5' to the 3', because if one strand of the DNA has the polarity as 5' to the 3', and another one will be with 3' to the 5'. Next, 16th question. Which nitrogenous base provides additional stability to the DNA? The nitrogen bases provide, which nitrogenous base provides additional stability to the DNA? The DNA is having two purines and two pyrimidines. In that, you all know that DNA is compared to the RNA, DNA is more stable. Why it is more stable? Because of the presence of thymine only in the DNA. Whereas in case of RNA, no thymine is present. Instead of thymine, there will be presence of the uracil. If the uracil again, if it changes its structure, or it is almost similar to the cytosine. So cytosine is said to be as the less stable nitrogen base. As in case of DNA, thymine is present. The stability of the DNA is more when it is compared with the RNA. 17th question. State the central dogma of molecular biology. The central dogma of molecular biology states that the genetic information flows from DNA to RNA and from RNA to protein. It involves the two important steps, that is called transcription and translation. When the genetic information flows from DNA to RNA, it is called as transcription, and if that information flows from RNA to the protein, then we call that process as a translation. Next, 18th question. What is reverse dogma of molecular biology? The reverse dogma of molecular biology is the genetic information flows from RNA to DNA. In the central dogma of molecular biology, the genetic information flows from DNA to the RNA. But in case of reverse dogma of molecular biology, the genetic information flows from RNA to the DNA. Next question, that is 19th. What is nucleosome? The nucleosome are negatively charged DNA which is wrapping around the positively charged histones and forms the octamer structure. I'll repeat, the nucleosomes are the negatively charged DNA which wraps around the positively charged histone octamer forms the structure called as nucleosome. Next, 20th question. Name the proteins which form the core of nucleosomes. As I said, in the nucleosomes there will be presence of histones, or we call them as the proteins also. So it is going to the histone octamer, I called it during the structure of a nucleosome. Why histone octamer? Because there are 8 proteins are present. Don't think that 8 different proteins are present. Only there are 4 histone proteins are present, where those 4 histone proteins are present in 2 sets and forms the histone octamer structure. So what are those proteins which are present in the core of nucleosomes? They are H2A, H2B, H3 and H4. 21 question. Mention any one basic amino acid present in large quantities in the histone proteins of nucleosomes. The large quantity of histone protein present in the nucleosome are, that is, the amino acid which are present in the large quantity in the histone proteins of the nucleosomes are lysine and arginine. 22nd question. How many base pairs of DNA helix are found in a typical nucleosome? So in a typical nucleosome, remember, there will be presence of 200 base pairs. Okay students, the remaining questions I will be discussing in the next class. Thank you.